Hello and welcome to today's webinar titled Applying Personalized Genomics to Ash Trees. This is a new topic for Frontline Genomics webinars, and I hope you're as excited as we are. Our main speaker for today is Richard Buggs, a professor of evolutionary genetics at the School of Biological Sciences at Queen Mary University of London and a senior research leader at the Royal Botanic Gardens at Kew. With an introduction as well from Sophie Zhu, account manager at Novogene, our sponsor for today's webinar. Before we begin, just a couple of quick announcements. First of all, questions. Please feel free to ask questions as soon as you like using the area on the right-hand side of your screen. You can also upvote other people's questions whenever you like. We should have plenty of time for Q&A at the end when we'll bring the speakers back on screen to answer your questions. On demand, we won't be sharing the slides from today's webinar. However, you can access the whole webinar again on demand whenever you like, within a minute of today's live session ending. Just use the same link that you used to get here today. You can also find all of our webinars on demand by searching on your internet browser for Frontline Genomics and Livestore, the name of our webinar platform. So let's start the webinar. I'd like to hand you over to Sophie, who's going to say a few words and also introduce our main speaker, Richard, in more detail. Over to you, Sophie. I'm just going to bring your slides up now.
Yeah, sure. Thank you, Richard Long.
There we go.
Yeah, I got it. Thank you. Hello, everyone. My name is Sophie, represent Novogene in UK. Before the webinar, I'm just going to give you a quick introduction about Novogene Europe. We founded in 2011, and our mission is to improve life research and clinical diagnostic research and human healthcare. Our work covers main aspects of all the next generation sequencing with wide range of application. Our lab is literally based at Cambridge Science Park, UK, and it's ISO 17025 certificate. We equipped multiple NovaSeq 6000, and we have got on-site superclustering computer, which means your data will be secured in UK securely. We have very strong global presence with 3 labs in China, 1 lab in Singapore, 1 lab in Cambridge, UK, 1 lab in United States, and we have an office in Netherlands. We provide sequencing services for both RNA and DNA. For DNA, we provide the sequencing services for human genome, microbiome, animal, and plant. And today, Richard is going to bring his science story in using our plant whole genome sequencing technology. So there are many sequencing companies in the market, and why Novogene? The number 1 reason is the quality. Our quality surpasses Illumina standard, and that's ready for the paper publication. The number 2 reason is turnaround time. Most of the project can be completed within 1 month. For whole genome, whole exome, and RNA sequencing, the turnaround time is 15 working days from sample QC to data delivery. In terms of the capacity, we are capable of sequencing hundreds of thousands of genomes per day. We've been sequenced over thousands of trees with Richard, and we've managed to deliver the work in time with high quality. Thank you for our clients. We are very proud of our clients like Richard Buggs. Therefore, our Novogene's name is shown in top-tier science paper with high impact factor. We have over 15 sales colleagues, and we speak over 25 languages. So no matter where you are based in Europe, your nearest account manager will come to you, talk to you, discuss your project in detail with your preferred language. So here we are. If you leave a message in our website, Twitter, or LinkedIn, email us. We will be in touch with you within 24 hours. So yeah, that's enough for Novogene. And I would like to hand over the talk to our today's speaker, Richard Buggs. So we've done very sequencing work with Richard. Richard Buck is the senior research leader at Royal Botanic Garden, Kew, and he is also the professor of evolutionary genomics at Queen Mary, University of London. So over to you, Richard.
Okay, Richard. I know, just to explain, I don't think Richard can hear or see Sophie at the moment, but if you can hear me, Richard, you're ready to go.
Well, thank you very much, Richard. Can you hear me?
Yes.
Excellent. Yeah, I'm sorry, I couldn't hear what Sophie was saying then. My connection's pretty poor. So I'll thank Sophie for whatever she said in introducing me, and thanks to Richard as well for hosting us here today. And I hope my connection holds up well enough for you all to hear me throughout this webinar. So I'm going to speak about applying personalized genomics to ash trees. It's not something you sort of expect to have personalized genomics applied to. Back in 2012, I was doing some work funded by the Natural Research Environment Council in the UK on birch trees in Britain. I had a fellowship from NERC to work on this, and then I became a senior lecturer at Queen Mary. And the first teaching that my head of department asked me to do as a new lecturer was to teach a course on human genetics and genomics to third-year biomed students. Well, to start with, I was a bit taken aback. I was working in the field of genomics, certainly, but not the genomics of humans. I was working on the genomics of trees. So to start with, I was a little bit disconcerted to be given this course. I found this excellent textbook exploring personal genomics, and sort of molded my course around that textbook, and of course did a great deal of reading in the primary literature, and came up with a brand new course on human genetics and genomics. Well, little did I know how much that was going to help me in my own research over the coming 9 years or so. And it has had actually a huge impact on my research on plants. And the reason for that is because most people working on plant genetics are working on crops like wheat and barley that are annual crops, or perhaps they're working on the model plant Arabidopsis. It's very easy to do. Well, I'm sure people working on them wouldn't say it's very easy to do genetics on them, but it's comparatively easy to do genetics on annual plants that produce lots of seeds, because you can very quickly do a cross and generate a family, and then from that family build a genetic map. With trees, you can't do that. A tree has a very long lifespan. It's often many years to get from seed to seed. It's very hard to tell trees who to mate with. It's very hard to do trials on trees. And in many ways, working on trees is very much like working on human beings. I'm sure many of you listening are scientists who work on human beings. And as with trees, with humans, you can't tell humans who to mate with. You can't just set up a mapping population of 2 individuals of your choice and ask them to produce several hundred progeny between them. You can't manipulate their environments and put them all in the same conditions, and they have a very long generation time. So in many ways, those of us who work on trees face the same kind of problems and challenges as people who work on human beings. And this is why doing a course for undergraduates on human genetics and genomics was actually incredibly helpful to me in my development as a scientist. Let's just have a look in more detail about why trees are difficult to work on, and then an unexpected bonus of working on a tree like ash. You know, if we compare wheat and ash, wheat has a generation time of less than a year, a time to harvest of less than a year. It grows in a small space. You can have inbred lines. With ash, long generation time, long time to harvest, huge space to grow, no inbred lines. However, the wonderful thing about ash is its genome size is only 0.9 gigabase pairs. It's, you know, less than a third of the size of the human genome. whereas wheat has a genome size of 17 gigabase pairs, much bigger, of course, than human beings. So there's a real opportunity here. We can do genomics quite cheaply on ash because of its small genome size. And this is actually true of most broadleaf trees. So here's a list of the main tree species that are grown in the UK with the hectareage of their growing. And Sitka spruce is the most grown one, but that has a genome size of 20 gigabase pairs. So does Scots pine. Most of the conifers have huge genome sizes, but you can see broadleaf trees like oak and birch and ash. They all have these really nice small genome sizes. So there's an opportunity here. And in the past in the UK, a lot of breeding effort and research has gone into these really widely grown conifer species. But if we think about the opportunities afforded by the small genome sizes of the broadleaf trees, genomics could really open things up for rapid advances in our understanding of the genetics and the genetic basis of traits in these important trees. And of course, we need our trees, and we're more aware of this than ever. Here's just a couple of BBC articles. Trees can solve global and local problems. And governments all over the world have pledged themselves to growing millions and millions of trees, huge tree planting programs going on. But of course, for these to be successful, we have to understand which trees are adapted to which environment. We need to know what tree to plant where. And we also need to know how those trees are going to resist pests and pathogens. And if genomics can give us some insights into this more rapidly than experiments, given that experiments are bound to last a very long time with trees, then we could really get some low-hanging fruit here and enhance our tree planting and tree growing activities across the globe. Pests and pathogens are of particular concern because the number of biotic threats to trees is just growing and growing. And unfortunately, largely, this is driven by global trade. Here's a map in the background showing global shipping routes. It's from shipmap.org. And it just illustrates the absolutely huge amount of global trade that's going on. And of course, a lot of this is in containers, and some of this is of live plants. And unfortunately, the list of pests and pathogens that have moved around the world and moved from host species, which can resist them really well, into naive populations that cannot resist them. The list of such pests and pathogens is just growing and growing. So we have a really urgent need to try to work out what makes trees resistant to pests and pathogens. And so that's what my work is focusing on in the case of ash. Now, ash is threatened in Europe by a fungus called Hymenoscyphus fraxineus, which causes ash dieback. And you can see a map here that shows its spread across Europe just over the last couple of decades. It was first found in Poland and then gradually spread across the whole of Europe. The emerald ash borer, thankfully, is not yet found in the UK, but in America, it has devastated ash populations. And both of these, this fungus and this beetle, both come from the far east of Asia and seem to be accidental introductions due to human activity. And we urgently need to discover how we can help our ash populations to resist these problems. I should also say that the emerald ash borer has been found around Moscow in Russia and seems to be heading towards Europe. So that's a very pressing concern for Europe as well, even though it's not here yet. So my group did a little bit of work quite early on in the ash dieback epidemic, looking at the mortality rates that we might expect for ash in Europe. And we looked at areas that had had different lengths of exposure to ash dieback and then fitted a model to predict mortality. And we found that plausible ranges of mortality were up to around 80% or more of ash trees could die from ash dieback. So this would absolutely devastate the landscape of Europe because ash is a very common tree. And also, if you just think about the implications for carbon sequestration, etc., it's a real live issue. Fraxinus americana in the USA was extremely common and is now listed as critically endangered simply because of the emerald ash borer. Well, my group got some funding from the Natural Environment Research Council quite early on to sequence the genome of the European ash trees for the first time. And we did this with a large number of collaborators. And we published that as our initial move into ash genomics. But we wanted to go from there to being able to predict how susceptible trees are to ash dieback. And we set up this website, Ash Tree Genomes, to host our work. And of course, everything is on the European Nucleotide Archive as well. So having published that paper with the genome, we then did a study on the genomic basis of European ash trees, European ash tree resistance to the ash dieback fungus. And you can just see some of the members of my group that were involved in this paper that was published in Nature Ecology and Evolution a couple of years ago. We were very fortunate in that in 2012 and 2013, Forest Research, which is a state agency for research on trees, set up some mass screening trials at 14 different sites within the UK. And they took ash trees that had been grown from 15 different areas within the UK and also from Germany, and planted them out in several sites in the southeast of England where ash dieback was already pretty bad. We focused in particular on two sites, North Birmingham and Frant Place, where there was good establishment of the trees and a lot of ash dieback damage. And here's a picture of what these trials looked like after a few years. Rows of trees evenly spaced, and some of them dead, others dying, and generally looking very ragged as the trees tried to resist the ash dieback fungus, which is coming in on spores from the surrounding area. We scored each of these trees from 1 to 7 by how dead they were. 1 to 3 are different ways of being dead, and then 4, 5, 6, and 7 are trees of different levels of health, with 7 being a completely healthy tree. And here's the results from just one of the sites. You can see the scores of 7, the 7 scores, that's the healthy trees, are very, very rare. Very few, a very low percentage of the trees are healthy. Lots of them are sort of in the middle, pretty unhealthy, and a lot of them are dead. So this was a great system to start looking at the genomics of resistance to ash dieback. Had very large sample sizes and good damage going on. So here's what we did for that paper. We took trees from these trials. We scored 38,000 of them. Then we took 1,250 trees, and we separated them into pools. Now, we would have liked, of course, to have sequenced the genomes of all 1,250 trees, but we couldn't afford to do that. But we worked with data from all of those trees by pooling them. First of all, we divided them into healthy and unhealthy in equal size groups, and then we divided them up by their provenance from the area of the country that they'd come from. So we had lots of different pools, and we sequenced the pools together. We had a mix of DNA, sequenced it all, and that enabled us to estimate the allele frequency of every SNP within those pools. And based on that, we could do a genome-wide association study, which is what I will show you about in a moment. Here's a PCA summarizing the variation in the SNP data among these different pools. You can see the different colors show different parts of the country. So red dots are trees that were originally sourced in England. So they're from a genotype from England. Blue ones are from Scotland. Yellow ones are from Ireland. Green ones are from Wales. And these two black ones off to one corner are from Germany. And what encouraged us when we saw this PCA was it to some extent seems to mirror geography. A bit like with human genetic variation across Europe that mirrors geography, our data seemed to. So this made us think that we hadn't done any messing up of mislabeling or anything like that. It was giving us a pattern that made a lot of sense. So with these different pools, we did a GWAS, and here's a Manhattan plot. And there were 203 SNPs that were significant at the threshold that we chose. So we were quite encouraged by this. And when we drilled down into what genes might be hit in these peaks in the Manhattan plots, some of them had previously been annotated by studies on plants like Arabidopsis as being genes that could plausibly be involved in pathogen resistance. So for example, here's one of our hits. It's an F-box gene, and in it we found one missense mutation, one mutation in a 5' UTR, and a couple of downstream mutations. So it was plausible that we'd found a biologically meaningful signature here. Here's another example of one of our hits, a protein CPR5-like, and that modulates salicylic acid, which is involved in plant growth and plant stress response. So we were encouraged by these results. Then we wanted to use them to do genomic prediction. For those of you who work on humans, this is a bit like doing a polygenic risk score. Genomic prediction is a method used a great deal by people working on crops and on farm livestock. It's good for breeding, for example, cows that produce more milk, etc. So to do our genomic prediction, we needed a test population. So we did individual whole genome sequences of 150 individuals, 75 individuals that were healthy and 75 that were unhealthy. And that was a different set of individuals to the ones that went into the pools that we used for the GWAS, but were being grown at the same mass screening trials. And all of them were from the same area of the country, were from just one provenance. So we then looked at how well we could predict, using our genetic data, how unhealthy the trees were. And this is quite a busy slide. I appreciate that. So I'll just walk you through it. The first thing to say is that the top row and the bottom row are essentially showing the same thing, but for different numbers of SNPs. We would have shown just two charts with all the different numbers of SNPs on all of them. But we just found that was too complicated to see. So you can see here that in the upper two panels, we're looking at a number of SNPs in the training set of 100, 200, 500, and 1,000. And in these lower panels, we've got training sets of 5,000, 10,000, 25,000, and 50,000 SNPs. And of course, we expect that the more SNPs we use, the more accurate our genomic predictions will be. So what we have here on the vertical axis is the correlation of our genomically estimated breeding values and the phenotype. And then along the horizontal axis, we've got the percentage of training SNPs used to calculate the genomically estimated breeding value. So this point here, for example, is using 1,000 SNPs in our training set and using all 1,000 of them in our genomically estimated breeding value. So that's the very tip of the green line in the top left-hand chart. Now you can see our correlations aren't particularly high. We've got correlations of sort of 0.2, and then in the bottom sets of charts on the left-hand side, with more SNPs, we've got correlations up to about 0.35. So that's not a very strong correlation, but for genomic prediction, that is quite a respectable correlation to get. And then if we look at the two charts on the right-hand side, this is the accuracy of our phenotypic assignments of the trees. So with a decent number of SNPs, looking at the bottom right-hand chart, with a lot of SNPs in the training population, we could get an accuracy of over 0.65. So what this means is, if you gave us a certain tree, we could predict from our genetic data how badly affected by ash dieback it would be, and 65% of the time we would be right. Again, you might say, well, that's not particularly impressive, given that, you know, if you just did it at random, you'd have a 50% chance of being right. But for genomic prediction, these are reasonably respectable numbers. And these mean that you could start using this data to inform a breeding program. And particularly, if we look at the top 20% of trees by genomically estimated breeding value. So if we just take the top 20% of our predictions and just the top 20% of the trees, the best trees, then our chance of getting them correctly assigned gets well over 80% if we use 50,000 SNPs. And so that you can see very easily how that would be useful in a breeding program. If we were to go out and do our predictions on a large number of trees and just take our top values, we could be pretty sure that we were getting a large number of trees that genetically have high resistance to ash dieback. So that could inform a breeding program. So we were quite encouraged by this. So as a next step, what we thought, well, let's try and roll this out to a woodland with ash trees of different ages growing in different environments. And we picked a woodland in Surrey called Marden Park Wood, which is run by the Woodland Trust. And it's an ancient woodland, lots of ash trees, lots of natural regeneration going on of those ash trees. A few different environments. Here's a photo of it. And excuse me. In a patch of this woodland, we mapped the ash trees. We estimated their size. We estimated their damage from ash dieback. And we took a sample for whole genome sequencing. And we had some hypotheses. We were hoping that, number one, frequencies of alleles at some loci will consistently co-vary with level of ash dieback damage, so we can predict the ash dieback damage of individual trees. So we can basically do personalized genomics for ash trees in a wood. And we hoped that many of the loci that consistently co-varied with ash dieback damage would be the same as what we'd found in the mass screening trials in our former paper. So perhaps we would even be able to predict the amount of damage each of these trees in the wood would have from the models we made from the mass screening trials. We also wanted to look at whether there were differences in the allele frequencies between the smaller, younger trees compared to the older trees, because we reasoned that ash dieback has been in this wood probably since 2012. And we know that ash dieback more rapidly kills young trees than old trees. So we expected that anything that had grown since ash dieback arrived may have been selected at a very early age for resistance to ash dieback, with those ones that are susceptible being killed off. And so we would see in the younger generation an enrichment for those alleles associated with resistance to ash dieback. And so basically we would see natural selection in action in real time in a natural population, which would be really cool. And it would also encourage us that in the long term, natural populations of ash would bounce back from the ash dieback epidemic, that even though lots of trees would die, the survivors would have enhanced resistance. And so the populations would be able to survive. Here's a map of the woodland. The different sizes of trees are shown by these green circles, and then small red circles are the very young trees, and small black circles are also very young trees too. And so you can see we've got lots of older trees and lots of younger trees. The younger trees tended to be in patches where natural regeneration could easily happen, and we got a good scattering of the older trees. So we sampled all of these, and here is a histogram showing the number of trees against the size. So we've got lots and lots of trees that are really small, and then lots of bigger trees, but really tailing off. And that's just the natural dynamic of the population. That's what was there in this patch. Here's our ash dieback damage scores, for the juvenile trees, they were quite easy to assess. And we used a health score of 1 to 5, with 5 being more healthy trees and 1 being unhealthy trees. For the big trees, though, we couldn't use that scoring system. We just had to estimate the canopy cover, assuming that trees with a full complement of leaves, 100% canopy, were healthy, and trees that had lost lots of leaves were unhealthy. Now, that's a pretty poor phenotypic measure. It's the best we could do, but there's many problems with this, because it could be that actually trees that drop their leaves really rapidly, if they get infected by ash dieback, are actually more resistant, because that could be a resistance mechanism. It's much better to be able to look at what's going on in the wood of the trees than what's going on in the leaves, which is what we could do with the younger trees, but not with the older trees, unfortunately. Anyway, so we collected DNA from 600 trees, and we sequenced them individually at 10x Illumina coverage, and we did that work with Novogene, generating between 4.5 million to 213 million reads per sample. And mapping all of that, we found about 21 million SNPs found in this population. And I must say, Novogene turned around this large sequencing project extremely quickly for us. We were under a lot of time pressure, and they did this work in a matter of weeks, which we're very grateful for. Here's a PCA summarizing the variation within the population, and the adult trees are shown in pink and the juvenile in this turquoisey color. And you can see already there's a slight difference in the variation between the adults and the juvenile trees. Then we tried to predict the ash dieback damage of the trees, and we did this using our mass screening paper data as the training population, and then using that to predict the amount of our genomically estimated breeding values for each of the trees in the population, and then doing a correlation between our predictions and the actual estimates of canopy cover that we took ourselves in the wood. And so here's the results for 69 adult trees. And unfortunately, it's not a very encouraging result. So we've got an R squared here of 0.0036. We are basically unable to accurately predict the amount of canopy cover on these trees under ash dieback using our training populations from the mass screening trials. We looked at the younger trees as well. We did genomically estimated breeding values for them, and we compared them to their health score. And again, unfortunately, we were not able to accurately predict the health of the trees based on those models. We're still working on this, and we want to try training the models based just on the data from this population. And we're also working on looking for differences between the old trees and the young trees. And here's a really quick look. This is very much a work in progress. We're still analyzing this data now. But this is looking across the whole genome, looking at SNPs that have increased or decreased in frequency in the younger generation versus the older population. And each point is a SNP, and the red ones are ones where the difference between the two populations seems to be significant, and that's with a p-value of less than 0.001. So you can see that at a quick first look, we are finding some alleles that have changed in frequency. Of course, we'd expect that just under neutral models, so we have a huge amount of work still to do. What we need to do now is predict the parentage of each of these young trees, and then compare different families within the population and see if there are particular alleles that are consistently missing in the families in comparison to the parents. So we're doing a relatedness study to try and map what are the parents of these different juvenile trees. So we've got a lot of work still to do, and we're having to think very carefully about why is it that we're not able to make better predictions of the health of these trees. And I think there are multiple factors going on. This wood has a different genetic background, perhaps, to the trees that we worked on in the mass screening trials. The trees in the mass screening trials were all the same age. They were grown in uniform environments with uniform spacing. And so environmental effects were definitely much smaller than the environmental effects that we're seeing in the wood. In the woodland, there's lots of different microenvironments, and these might be having a huge effect on how much inoculation each individual is getting from ash dieback, and also their general health and their general ability to resist ash dieback. So it's all extremely complicated, and I'm sure those of you who work on humans will appreciate how difficult it is to get uniform environments with studies like this, and to actually make accurate predictions based on genomic data. But as I say, we've only just started analysing this big data set, and we still have a lot more work to do. So we're very much hoping that we can come up with some better predictions as we drill down further into the data. But what we're finding overall is doing personalised genomics for trees is complicated and difficult. And we knew that was the case for humans. It's also the case for trees. Well, I'd like to thank the postdocs and PhD students who have been involved in this work on ash trees and ash dieback. I haven't had time to talk about some of the work we've done on emerald ash borer as well. And we also work on oak and birch. I've just talked about ash trees for the purposes of this talk. And this work has been largely funded by the UK Department for Environment, Food and Rural Affairs and the Natural Environment Research Council. So I'm very grateful to them for their generous funding for this work. Thank you very much.
Thank you so much, Richard. Just what a brilliant presentation and sort of laying out of your research. Really appreciate it. I know the audience have too, because we have some questions. Let me start with a question from Abhishek Gogna. How did you select the SNPs used for genomic predictions, and how were the accuracies on that same slide defined?
Great, thanks. Well, we would have liked to have done our genomic predictions on all of the SNPs we had, but we had many millions of SNPs, and we simply didn't have the computational power to use all of them. You know, often people will be doing genomic predictions based on an array, so they already just have a few tens of thousands of SNPs, whereas we literally had millions, because we were doing this based on whole genome resequencing. So what we did as a sort of way of whittling down the number of SNPs was we took the top hits from the GWAS. So we took the top 50,000 SNPs according to the GWAS, according to their effect size, and then we used them to do the genomic prediction. And then for the smaller sets, we used, you know, things like the top 200 from the GWAS. Of course, that's not a perfect approach, because just because something is a hit on a GWAS doesn't mean it has a large effect size. It doesn't mean it will necessarily be useful in genomic prediction. But we thought that was the most sensible way of narrowing down the number of SNPs we had. There may have been a better way, but that's the way we took. The accuracy was, once we had done our genomically estimated breeding value, let me just pop back. Basically, we assumed that well, we had to define a cutoff, of course, which was tricky. So did we predict something was healthy if it was in the top 50% of our breeding values, or did it have to be in the top 80%? And to be honest, for this one, I would have to go back and look at the paper. For this one, we said, okay, we'll just predict that it will be healthy if it's in the top 20% of our genomically estimated breeding values. And then if our prediction is correct, then great. If it's not, then we were wrong. Is that an adequate answer to that question?
I think so. I know Abhishek may leave a comment to let us know in the next few seconds. If there's any follow-up to that, Abhishek, please let us know in the chat or the questions section. Yeah, Abhishek saying yes on the chat, just in case you can't see that. Brilliant, thank you. A question from Katie O'Brien. What software did you use to train the models?
I'd refer you back to the paper for that. So this was all done by Carey Metheringham in my group. I know for the GWAS, we used some software called Populations, with the word pool incorporated in population, because it was specifically designed for doing on pooled sequencing. I have a feeling we used RR-BLUP, but please do go back to the paper and check, because yeah, that's the most accurate representation of what we did.
And I think, and I might be wrong, but I think this research has been detailed in a few different publications. Is there a particular paper that you would direct people towards?
Yeah, so there's the paper Stocks et al. In Nature, Ecology and Evolution. That's where all the details for this are found. So the GWAS and the genomic prediction are all in this paper here. That's the place to look.
Perfect. Thank you very much. Another question from Niels Muller. In the woodland individuals, did you specifically look for allele frequencies of the promising candidate genes found in the mass screening?
We haven't gone through the top GWAS hits one by one and looked for changes in allele frequency as yet. All we've done so far is try to predict their health based on a model that we trained on the mass screening data. And we've looked genome-wide at changes, but we have a long list of things we want to look at that we haven't yet had time to look at. Very much a work in progress.
Thank you very much. Another question from Pablo Noceda. How many trees did you pool for sequencing?
Each pool was 30 or 40 trees.
Brilliant, thank you. A follow-up question from Abhishek as well, saying, but you did not mention the model used for GWAS. What effects did you model?
Again, I'd refer you back to the paper for all of the details. If I try and do it from my recollection, I may well mislead you.
Brilliant. And just for those of you that do want kind of easy access to that paper, obviously the details are there, but Katie O'Brien has very kindly dropped the link into the chat tab as well for you to just kind of click through. A question from Rena. How did you measure the canopy cover percentage?
Yeah, good question. Unfortunately, we just had to do it by eyeballing it from the ground. So what we would like to do in future is use drones to do it. And we've started in collaboration with a PhD student in geography at Queen Mary. We've started to do some drone work on the trees, but we haven't yet got to the stage where we can use that to do a really accurate measure of canopy damage. But that's where we're trying to head with this work.
Brilliant, thank you. And a question from Amy Baldwin. Does genetic diversity in the pathogen population, and I'm afraid I'm gonna ruin this name if I attempt, I'm gonna attempt it anyway, Hamenocyphus fraxineus, that might make a lot more sense to you. Does that have any effect?
Well, the population of Hymenoscyphus fraxineus in Europe has quite low genetic diversity. It was the paper on the genome of the pathogen was published a few years ago, and they think that there was a bottleneck of 2 when it colonized Europe. So it has relatively low genetic diversity. Of course, 2 individuals can contain within them a huge amount of diversity, but it's because it's come from 2 individuals and rapidly expanded. We think that probably within the areas where it's rapidly expanded, there's probably not much genetic structure within that diversity. So we're not expecting that to have made a big difference. But it you know, it might be something worth looking at.
Thank you. And a question from Fiona Cork. Have the healthy trees, in inverted commas, in the training set been rescored for their health recently? Do they show delayed disease susceptibility?
We have gone back and rescored them. And generally there is consistency between the years, but sometimes a tree is very much worse off, and occasionally they seem to bounce back as well. So this is another complexity of the scoring, that the changes you see between years in the trees.
Brilliant, thank you. And then at the moment, it's a final question, I think, from Paolo Lorenzini. Do you think that your prediction method can be applied to crops as well in a more straightforward manner, considering that in many crops we have already well-studied SNPs stroke genes related to abiotic and biotic stress resistance? And a follow-up, how would you further improve accuracy?
Sorry, I didn't catch that very last.
The back end of the question was a follow-up. How would you further improve accuracy?
Great, yeah. Well, genomic prediction is being done on crops already, to some extent. I guess what's slightly different about what we've done is that we use pooling, and also we are just classifying trees as resistant or susceptible, which in some ways is not ideal. What we're trying to do is come up with our own method of doing the genomic prediction, and Kerry Metheringham is working on this at the moment, actually, coming up with a more bespoke genomic prediction method that takes into account our pooling and the way we've scored the trees, and that's something that we're looking to publish on quite soon. And of course, we're hoping that might have a more general utility.
Brilliant. I think that's all for questions, unless there's more wanted. No, just people saying thank you. Great talk, lots of those. Yeah, thank you very much, Richard. I know you've been battling a cold today. I thought you've held up really, really well. Really appreciate you taking the time out to present your research. Thank you also to Sophie as well for that great introduction, and also to our partner for today's webinar, Novogene. I think that's all we have time for today. Don't forget, this webinar is available on demand, so you can watch it back at any time. And that's pretty much it. Have a lovely day, everybody. Goodbye.
Bye.